So hello and welcome to another class of AB Sciences. This is Abhishek with you. So today I'll be talking about the elongation phase of the protein synthesis that I have already told you in the last class. I have completed the tRNA charging part, amino acid activation as well as initiation. So I will start today with the picture which I have finished that day. So it will be easy for you to understand if I draw some diagrams. That's why to save time I have already drawn it. So here you can see this is the ribosome, this red one, 70S ribosome. Now as you can understand, this is the mRNA 5' to 3'. This one, this is the tRNA molecule. I have drawn it to save time and to make you understand only. And this red dot or the circle is the amino acid, that is the tRNA is charged with the amino acid. As you know from the previous class, this is the tRNA F-met, which is charged with formyl methionine amino acid. So it is the initiator tRNA with initiator amino acid that is already present, as you have seen in the initiation complex. Now, as I told you that day, that the next amino acids which will come will be binding towards the A site, that is the aminoacyl site. So now, suppose one amino acid is coming, that time it doesn't attach themselves, or the ribosome cannot just add it. It needs energy, and the energy comes from GTP. So the few factors which we need here is EF-TU, EF-TS, GTP, and EFG. So these few things we need here to proceed with the elongation process. Now, initially, that I have seen in the initiation, IF2 GTP complex is required. Now here it is EF-TU GTP complex is required. EF-TU GTP complex. Now this EF-TU GTP complex recycles with the help of EF-TS. So if I draw some circles like this blue one is the EF-TU, red one is the EF-TS. Then in the middle, when it is bound to EF-TU GTP, what is bound? That is the tRNA with the amino acid. So I'll draw it here only, so it will be understandable. Suppose the next one is the CAA. So here it will be GUU, and the charged amino acid is here, and suppose the bond is GTP bond is here. This is the GTP bond. Now this tRNA, which is the next tRNA, which is going to bound to that CAA codon which is present in the mRNA. Now here EF-TU GTP complex will come and bind with this codon in the A site. So already we have UAC with the first amino acid. Now the next amino acid will come with the anticodon of GUU, and here it is the amino acid. So this EF-TU GTP and EF-TS GTP complex is just a cycle which is going on and on till the elongation process is going on. Okay. Now here EF-TU and EF-TS actually they made a in-between structure when it is not bound to the GTP. Suppose EF-TU GTP is now bound, then it will hydrolyze with EF-TU GDP. In between, EF-TU will be bounding to EF-TU with EF-TS. Now this is the mediator phase. After that EF-TS GTP, that here EF-TS GTP will transfer the GTP group to the EF-TU, and again the EF-TU will give you the So I will remove this one, it will make you confused. So here EFTU GTP, now it is hydrolyzing, then EFTS GTP is coming, EFTU to in between phase is there. Now EFTS will move away with the GTP. So here you can write EFTS GTP, and the EFT GTP will come in the scene again. So this is the whole cycle which is going on and on during the elongation phase. So this is up to the factors. I'm not discussing again with it. Now come to the main point. So here you can see, as I have told you earlier, exit site is present in the 50S, 30S contains 50 and 30S contains both P and A, but E is exclusively present in the 50S ribosomal site. Now the two amino acids are bound to their codons with the anticodon. Now the next phase is the peptide bond formation. So the peptide bond formation is a critical step where one amino acid is joined with the another amino acid present in the tRNA. Now this formation of peptide bond is done by 23S rRNA, also known as peptidyl-transferase. Okay. So now I am showing the step of peptidyl-transferase formation. So I'll draw a quick picture. Formyl methionine group. The formyl methionine group from the first tRNA is transferred to the second tRNA which is present in the A site. Okay. So try to understand that the first tRNA, the formyl methionine group from the first tRNA is transferred to the second tRNA which is present in the A site. Now the alpha amino group of the second tRNA, alpha amino group of second amino acid, or you can say the second tRNA with the charged amino acid. So second amino acid, the alpha amino group, will act as a nucleophile and form the bond with the help of 23S rRNA, and the initiator tRNA, F-met tRNA, will now uncharged. So it is now uncharged without any amino acid, but the location is same, that is the P site. So P site contains now the tRNA, F-met tRNA, which is now uncharged with the amino acid, and that amino acid has been transferred to the second tRNA containing the amino acid and joined with a peptide bond, which is formed by the 23S rRNA, that is peptidyl-transferase. So up to this is the part of the elongation. So now the ribosome has incorporated a new amino acid, and the P site contains the uncharged tRNA, and this site is a dipeptidyl site now. Why it is dipeptidyl? Because two amino acids are present in the aminoacyl site now. Now there you can see there is no site here to be bound again. So the amino acid needs to again translocate, or it needs to transform from one region to another, or you can say walk. It can walk from one codon to another codon. So the ribosome, mechanism of ribosome is to walk from one codon to another codon. Okay. So here when it is walking, that is called the translocation. Translocation of the ribosome on the mRNA, and that is called translocation, which is done by the EFG GTP complex. So for translocation we need again GTP, that is the energy. Without energy ribosome cannot walk on the mRNA or cannot relocate from one codon to another codon. So if I draw again the next part of this next scenario, so look again, this is the picture of the next step after the peptide bond is formed and the dipeptide is present in the A site. This is up to this elongation has started. Now when elongation is proceeding, it will walk through the mRNA like one codon to another codon. I said already. So here you can see the scenario which will happening or which is happening inside the prokaryotic cell, where the E site, now you can see the UAC, that is a trans FMET tRNA FMET, is now transferred to the AE site. Okay, that is the exit site. So it will be removed or it will be transferred to the cytoplasm again for future use. Okay, and the E site will be empty again. And now which was in the PA site, the dipeptidyl tRNA, which is present in the A site, now transferred to the P site. Now this translocation is done by the EFG GTP and it will be released as a EFG GTP. Now after translocation, again EFTU GTP with another tRNA, charged tRNA, particular charged tRNA will come and again bind to the TTA, which is a codon here, and against this AAU anticodon, completing tRNA charged against the particular amino acid will come in the scene, and again in the same mechanism the 23S rRNA will act as the you can say the self-catalyzing enzyme here, you can say, and this will form the peptide bond. So likewise it is just ongoing, going, and at last it will encounter a termination codon or somehow other factors may be there to stop the elongation process. So I will tell you the termination in another class. Today the elongation is very important part of the protein synthesis, where you can understand the mechanism. Now here also protein synthesis fidelity is very essential. Now how this cell is maintaining the fidelity of the process? Fidelity means the error rate. So how it is reducing the error rate? So during EFTU to EFTS GTP to GTP transfer, that time the process is little bit slow. So if the GTP which is used here is not the GTP but the analog of GTP, suppose GTP gamma S, if this GTP is used instead of the original GTP or the actual GTP which should be there in case of EFTU GTP, that time the dissociation is very slow, and that is the time when the fidelity or the error or the proofread is going on, and this likewise it is proofreading the elongation process also. So all these mechanisms are there to reduce the error rate of protein synthesis. Now I hope you can understand. If you have any questions, please ask me in the comment section, or you can mail me also. Thank you. So if you like my classes, give a big thumbs up and don't forget to subscribe my channel, because these things stimulate me to take futuristic classes for you guys. So thank you and be with me.